Good morning, everybody. Thank you for joining us for the first Asthma Grand Rounds of the academic year. It's good to virtually see everybody again. And we are really looking forward to a talk by Quan Lu. He's the professor of medicine, Department of Environmental Health and Molecular Metabolism at the Harvard T.H. Chan School of Public Health. He is a Seltzer and Philip Drinker professor at the School of Public Health. And he's going to be talking to us about exploring the genetic basis of asthma. You can submit questions to our speaker by entering them into the chat. And for CME credit, please email to your name and your degree to eIsrael assistant at bwh.harvard.edu. If you want to shorten it a little bit, you can do eIsraelassist at partners.org. That will work as well. And for any technical problems during the broadcast, send an email to bwhebcast at partners.org. So bwhebcast at partners.org. And so, Dr. Lu, thank you again for coming to speak to us. We're looking to an exciting talk about exploring the genetic basis of asthma.
Great. Should I start?
Yes, please.
Great. Good morning, everyone. So thank you, Chris and Elliot, for the invite and for the kind introduction. So I'm going to share my slides. Let me see if I can do it. Can people see my slides?
Yes.
Great. Good morning. I give a rather broad title, Exploring the Genetic Basis of Asthma. I add a subtitle, Gasdermin B and Paroptosis. So today I'm going to talk about the work in my lab that exploring the role of Gasdermin B and Paroptosis in asthma. So I do have a disclosure statement. So I co-founded the two biotech startups over the past few years. Although the work I'm going to talk about today is not related to the startup activities. So for those of you who don't know me, I want to give a very brief introduction into the work in my lab. So I actually is a molecular biologist with training in functional genetics. So I was actually hired into the Department of Environmental Health at School of Public Health back in 2007. The idea was actually, I never worked in anything related to environmental health or lung biology or public health for that matter. So the idea was for me to bring my training in functional genetics applied to the study of environmental health and lung biology. So in the first few years in my lab, I was able to develop research programs, two research programs. One focused on metal toxicity because I'm in the Department of Environmental Health. There's a lot of interest in how metals can contribute to the many environmental diseases, including neurodevelopmental and neurodegenerative diseases. So I focus on arsenic and lead. And the other program I developed because I was hired as an assistant professor of lung biology. So I did actually develop a program focused on asthma biology. And again, using my training in molecular biology and genetics. Initially, I started a receptor signaling, in particular, the beta-2 receptor, and how the beta-2 receptor respond to beta agonist in the lung, in particular, the airway smooth muscle cells. Later on, I began to my interest in the really genetics of asthma, and that's the focus of my talk today. So interestingly, actually, my work in the looking at the beta-2 receptor signaling in the airway smooth muscle cells actually led sort of through serendipity, actually, the finding of a new type of extracellular vesicles, and that's kind of biologically very new, and we have been actually my lab had devoted much of time over the past few years to understand the biology, in particular how the extracellular vesicles that we discovered are involved in intercellular or tissue-to-tissue communication. And so it turns out that even though extracellular vesicle EVs are so different from my original study on matter toxicity or asthma, it turns out that they now actually they contribute to both programs, because extracellular vesicles have been found to be also important for neurotoxicity and asthma and lung biology in general. But today I'm not going to touch much about the EV work. So I would focus on here the asthma genetics. So for this audience, I don't have to convince you that asthma is an important disease, and it affects millions of people around in the U.S., around the world, and many of them are children, and there's no cure for asthma. So asthma is a classic a quote complex disease, because it's both genetic and non-genetic factors contribute to the pathogenesis of asthma. And non-genetic factors such as air pollution, exposure to allergens, viral infection, and even obesity can contribute to asthma. So very complex. But if you look at here's a sort of cartoon drawing of airways. On the left is a normal airway, on the right is asthmatic airway. You can clearly see the difference between the normal and asthmatic airway. And in the normal airway, you have a very open and clean airway. You have an airway wall wrapped around by this airway smooth muscle cells. But in asthmatic airway, you can see that there's a lot of changes. So there's a mucus buildup in the airway. The airway wall thickened, and there's inflammation, and the airway smooth muscle cells in asthmatic airway is much tightened and constrict. And as a result, the airway is become narrow and with limited airflow. So if you do a section of this normal asthmatic airway, and you can see the more detailed structure changes. Again, on the left is a normal airway, on the right is asthmatic airway. Again, in the normal airway, you can see the epithelium very nicely lined up, and then you have some smooth muscles as this pinkish staining here, and the airway is open and clean. On the right, the asthmatic airway, a lot of changes, structure change. You can see that the airway epithelium become very buckled, and there's sort of squeezed, and a lot of mucus secretion build up from the epithelium. And then the big change in the base membrane, in particular this airway smooth muscle band, as you can see, dramatically increased, and even the blood vessels seem to change. So there are a lot of changes here, and everybody agrees that these changes occur in asthmatic airway. But the big question is that what actually caused these changes? What lead to this nice open airway become this constricted and narrow airway? And so I think that people study epithelial cells or smooth muscle cells may argue that it's because this changing in these cell types, the biology, changing the cellular biology lead to this change in asthmatic airway. And immunologists might say, well, this is probably because of the aberrant sort of immune response to the exposure to these non-genetic factors, air pollution, allergen. A person who study biomechanics in the lung might argue it's because the pressure, the mechanical structure changes in the airway that lead to asthma. So really that I think there's a lesser agreement on what really causes asthma. So to me, I think that really to find out the root cause of asthma, you have to go back to genetics, because we know there's huge contribution of genetics to asthma. But as I already said, the asthma is a rather complex genetic disease, because there's no mutations that contribute to asthma, unlike these classic genetic diseases where mutations dominate the pathogenesis of disease. So back in 2007, actually, exactly the same year as I started, a number of groups in here that Bill Cooksen's group identified actually genetic basis of asthma, where they did this so-called GWAS studies, genome-wide association studies. So for those of you who are not familiar with this, so this is on the x-axis, it are the genes on each of the chromosome. So you look at in these studies, they look at the genetic variants, not mutations, but variants that in this case, SNPs, single nucleotide polymorphisms. And then, so each of these dot, the circle dot is a variant. So each of us have a lot of variants in our genome. And these studies actually look at the contribution of these variants, and in fact, the association of these variants with certain disease. Here, look at asthma. So on the y-axis is a minus log p-value. So the p-value, the smaller, the higher the on the y-axis. So basically, if you have a very high value here, means that the association of with the disease is very significant. So this is also known as a Manhattan plot, because it does look like most of the buildings here and then some building very high up. In this study, Cookson group identify several locuses that with genetic variants, SNPs, that highly associate with asthma. And in this particular one, I highlight here, 17q21 locus. As you can see, there are multiple dots, meaning multiple variants that significantly associated with asthma development. So this finding actually is very exciting because it does actually confirm that what we already know, that asthma is a genetic disease. But again, there's no mutation. These are all variants. There's the effect size is relatively small. But this 17q locus is one of the probably the most reproducible and robust signal for, in fact, probably any genetic disease, but in particular here is associated very specifically with asthma. So if you look at so this study has been replicated in many other populations with larger population size. So it's the signal stands. And if you look zoom into the region, so as you can, again, this is a study by Carol Uber's group, where they look particularly in that 17q21 region. As you can see here, there are many SNPs variants that significantly associated with asthma again. But for biologists, we want to know actually where these variants are in terms of their genes. So you can see in this region, there are actually 6 genes here. So, and even though most of the significant variants occur in this ZPBP2, GSDML, actually also known as GSDMB, and ORMDR3, and a little bit GSDM1, also known as GSDMA. So the people are excited. These variants are clearly reproducibly associated with asthma, but which gene actually is involved here? So you can say, well, people may argue, well, I like this gene or that gene, but I think that I'm not going to speculate why people pick one gene over the other. But I think in the early days, most people focus on this gene, ORMDR3 here. I think because there's a homolog in the mouse genome, and also there's actually the yeast gene corresponding to this, and it's important for the sphingolipid biogenesis. And there's some study that links this gene to to asthma-like phenotypes. And some of the SNPs actually contribute to the associated with the different expression of these genes. But again, most of the genes actually, SNPs here identified are non-coding variants. So they affect gene expression. But again, that because they're sitting on the genome, so they could affect the neighboring genes. So really, it's very hard to say this is a gene, whether it's ORMDR3, GSDMB, or ZBPP2. It could be even more distant genes here. So really, to take a unbiased approach, and we saw that because most of the variants are non-coding, they could associate with more neighboring genes through the genome structure regulation. So we saw why not look at the coding variants, those variants that actually change the amino acid of a protein encoded by a gene, and we saw those are more likely to be functional because they change the composition of the gene product, the protein. So we took approach that very naive. We just said, well, we're going to look at all the coding variants in this locus and see how they associate with asthma. So that's what we did. So again, we look at the amino acid changing coding variants in the 17q21 locus. And we basically look at this a database known as Exome Aggregation Consortium. So basically, they list all the variants occur in the exomes, which is more likely to lead to the amino acid change because they're exons, not in the introns or intergenic genes in the regions. In total, we identified 8 coding variants with that are common, meaning that their minor allele frequency greater than 5%. Surprisingly, actually, we did not find any coding variants in 3 of the genes. So remember, there are 6 genes in that region. Or including the variant that most people study, ORMDR3, there's no coding common coding variant. It doesn't mean that the gene is not important, but in here we're focusing on something that more likely to be functionally associated with asthma. So if you look at these 8 common coding variants, and in this work it's a collaboration with Scott Weiss and Wu at the Channing. We look at the JERA cohort and 8 variants, and the top 2 variants. Obviously, you look at the minor allele frequency. Actually, it's very common. All these like half almost, like 45% is minor allele frequency, meaning that major allele is 55. So basically, it's a split. Some people have half of people with this minor allele, half of people with common. And then odds ratio is not super great, but it's very consistent because look at the range. It's the p-value is good. So again, the top 2 both coding variants occur in this gene gasdermin B, which is next to ORMDR3. At the time, not many people studied the gasdermin B, but now I think there's more and more reports and linking these genes to asthma. So the first thing we did at the time, there's not much known about what gasdermin B gene or protein does. So we saw that first look at the expression of this. So this is a work by Ron Pagniban and Mao Yun-sun. They collaborate with Gina Park's lab, which provide us with airways epithelial cells that culture from air-liquid interface, or ARI. So if you look at the expression of gasdermin B, smooth muscle cells don't express it. Fibroblasts don't express much, and then even in undifferentiated HBE cells, human bronchial epithelial cells, they don't express much. Only they expressed when they are actually differentiated through this ARI culture process, and you can see actually it's a really dramatic increase of the gene expression. And in these differentiated cells, which they differentiate from basal cells into ciliate cells, goblet cells, so we actually did the sorting analysis, sorted out ciliate cells versus in goblet cells. As you can see here, FOXJ1 is a marker. You can see the sorting looks pretty good. The tubulin 5 negative cells, they don't express FOXJ1. These tubulin 5 positive express a lot of FOXJ1. And the same thing for the goblet cells. Mucus 5 AC is a marker. You can see that you can sort out the either the non-goblet cells versus goblet cells. So if you look here, that in the ciliate cells here, actually the gastromin B expression is higher by twofold, not very dramatic, but there is a significant difference. So suggests that gastromin B may be more preferentially expressed in the ciliate cells, although it's also expressed in the non-ciliate cells as well. While the goblet cells versus non-goblet cells, there's not much difference here. But again, in this in the ciliate and goblet cells, the expression level is pretty decent. And we did the immune staining, the immune fluorescence staining, as you can see here, using cultured human airway epitheliums. This is green, the gastromin B. The antibody is not great, but they did stain the cells here and then colocalize decently with the beta tubulin 4 marker, which is for the ciliate cells. But again, it also expressed non-ciliate cells. We also did that with the section of human airway tissue. Again, gastromin B here. This is a beta tubulin 4 for ciliate cells. Similar conclusion. It is expressed in the ciliate cells, but not exclusively. So what at the time, actually nothing, that was 2014 or 15, nothing much known about the gastromin B's biologic function. In fact, so this is, and then in 2014, 2015, a group in China, Fan Shao's group in the National Interest of Biological Sciences in Beijing, actually identify a role of gastromin D. So again, this is a human gastromin B gene. So this is looking at the evolutionary tree of all the gastamins in in different organisms. This is the gastromin D. They're not very close, B and D, but so Fan Shao's group published a landmark paper in Nature, identify gastromin D actually is a gene, is a protein in major mediator of the process known as pyroptosis. So pyroptosis is a process, it's different from, it's basically cell death, but induced by inflammation. So it's a pyro, as you know, it's a fire. So basically it's a inflammation specific program of cell death, different from apoptosis, different from necrosis. So this is a review after the Fan Shao's initial publication, and this is show that actually gastromin D, not B, gastromin D here that can be cleaved by these inflammatory caspases such as caspase 1, 4, 11, and into both N-terminus and C-terminus. So N-terminus actually one after cleavage aggregates, form this beautiful pore structure on the membrane. So the pore size range from around 20 nanometers. It's very large pore size, allowing a lot of stuff from the intracellular content get into the extracellular space, and this causes a lot of problem, inflammation. So this study linked to the pyroptosis. So we saw that, well, at the time, gastromin B is not known to act like this. So we saw maybe the easiest hypothesis that gastromin B function just like gastromin D, could be cleaved and then form this pore-induced pyroptosis. So we did that. This is a gastromin B protein. We tagged with a flag tag and then test whether the protein can be cleaved by caspase 1. So Y type here, as you can see here, and then if you co-transfer caspase 1, you can see actually the protein gastromin B gets cleaved into the two small band. So this is a non-specific band in the middle, but this is a two cleaved band. This is a smaller product, likely is the C terminus because the prediction is here. And so in the middle, we look for potential the cutting site by caspase 1. So caspase depend on the this aspartate D amino acid. So there are two aspartates in this region. So we mutate either one of them to see which one is the cutting site. As you can see here, this site 236, if you mutate to the alanine, as you can see here, it's still expressed, but no longer can be cutted here. So as you can see here, you don't see the small cleave, the gastromin B. But if you mutate here, 250 site, it doesn't really matter. Same with Y type. So this experiment suggests that actually caspase 1 cleave gastromin B at this site, recognize this aspartate. So I thought this is great. And then we want to see whether the N-terminus, like in D, N-terminus goes to the plasma membrane and then wreck the whole thing up, and then just destroy the cells through this apoptosis process. So we just co-transfect either the control EGFP here, full-length gastromin B or N-terminus or C-terminus. As you can see here, only N-terminus transfection lead to the drastic cell death. As you can see here, same thing here. You look at the on the microscope, the cells just it's a massive difference actually. Only about 6 hours after the transfection, you can see the. And if you express the here Y type with the caspase 1, you can see the increased toxicity. Again, indicating that caspase 1 probably cleave it. If you have the mutation, as I said, which cannot be which destroy the recognition site by caspase 1. Even you co-transfect with caspase 1, there's no change in the cell toxicity. While if you this one, it doesn't really care. So again, indicating that cleavage by the caspase 1 of this gastromin B lead to the cell death. So we thought this is great. This paper, this data were included in this publication in early 2018. But then right after that, actually, there are multiple papers came out just said that we were wrong, because they don't see any evidence that the N-terminal gastromin B can induce apoptosis. This is one of the papers actually. So if you can see here, they express in gastromin B N-terminus, either here or this gastromin D N-terminus here. And they only report can see D can N-terminus can induce apoptosis, but not any of the B form. And again, this is there. So we sort of we know we're right because we see it. So turns out that if you look at actually how they did it, they fused this protein to the EGFP because that's easy because at the time there's no good antibody against gastromin B. So and GFP give you ability to look at the localization, and easy for transfection. Turns out that and they put the EGFP at the N-terminus of this gastromin B. And turns out that it actually destroyed the ability of N-terminus to induce apoptosis. We actually, in our experiment, if you put the flag tag at the N-terminus, we also do not see apoptosis. So it's very critical to not actually tag the N-terminus. So we know that, but these people, again, publish, so they're create a lot of problem for us because later on when people either they cite us or actually many people don't cite us, but when they do, they always mention these papers. Well, there's no actually evidence that the N-terminus induce apoptosis. And but we know we're right, and then the field actually realized that we're right because in fact this paper in 2000 last year show that gastromin B can be cleaved into this N-terminus and can induce apoptosis. Very, now again, they did not use the tag of the N-terminus. And this actually is a structural paper showing that N-terminus of gastromin B form this beautiful ring, sort of a pore on the plasma membrane, and so that's settled. Now it's known that gastrin B can induce very potent paroptosis. Now back to the asthma association. So again, as I mentioned, that we identified two coding variants in gastrin B gene. One of them is this proline to serine change here in this exon 9. The other actually is a splicing variant. So basically, this variant in here, if you have this Y type or major allele, it lead to this protein that's about 416 amino acid. But if you have this variant T2C here, splicing variant, actually it leads to the skipping of this exon because it's a splicing variant. It provides a splicing acceptor site. And this variant destroys the acceptor site for splicing. This eventually lead to the shortening of this protein, the segment here of 13 amino acid here. So there are two forms, 416 versus 403, depending on the splicing variant. So again, this show here, major form, minor allele form. You can actually, this is a human HBE cells. We actually genotype many of them, identify the one with major allele here, and you see this sequence with 3 exons here, including exon 6. But if you have a minor allele here, the exon 6 is missing. And then you can see the Western blot here. And in the major allele one, you see both variant, but in the minor allele homozygous, you do not see the 416 form. So when you cleave 416 into the N-terminus, it's lead to the fragment of 232 amino acid. If you express in the cells, massive paroptosis. But in the variant that with a shortening of the exon 6, you express this fragment, no paroptosis. It's very clear, day and night. And the same thing here, you express this 416 form with caspase 1, you have paroptosis, and 403, no paroptosis. We also include the other coding variant. It did not seem to affect the paroptosis much. Maybe a slight decrease, but not significant as a splicing variant. So this is all the data were published in this paper, and basically show that in the white, I wouldn't say white type, but in the major allele one, you have this 4-length protein, and then when it's cleaved, gets into on the membrane, paroptosis leading to the release of many molecules, including cytokines. But if you have the minor allele, particularly the homozygous for the minor allele, this leads to the skipping of this exon, smaller protein, and then when it's cleaved, it cannot form the pore on the plasma membrane. And from this, we hypothesize that the gastrin B induced paroptosis actually compromise integrity of all these epithelial cells, airway epithelial cells. This potentially lead to the inflammation and structural changes in the airway. That's asthma. So that's our hypothesis. So now we figure out the sort of genetic component or the molecular pathways here. We wonder, because the asthma is a complex genetic disease, there's contribution from environment factors. So we want to see what about the environment factors? How do they contribute to this pathway? So we already know the many asthma triggers, such as rhinovirus infection, exposure to allergens such as house dust mite and other allergens. So how do they fit into this pathway? So again, this is a work by the Ron Pagniban. So here he collaborated with a Jinnah's group again, using the ARI HBE cells, expose them to rhinovirus A, which are known to associate with asthma development in children. And if you look at the expression, gastrin B expression actually is pretty dramatically increased, particularly at the 24 hours after the infection. There's a dose response to the amount of virus you use. But if you look at the cytotoxicity, by the LDH release, we didn't see much change even in the higher gastromin B expression. So this suggests to us actually just increase the expression of gastromin B is not enough to induce proptosis, because this protein need to be cleaved. So we initially showed caspase-1, but caspase-1 is not very specific. It does not cleave gastromin D or the other gastamines. No, it does cleave. Caspase-1 is not specific, cleave all the gastamines. So is there anything that may cleave gastromin B? And so we know that there are a lot of proteases in many asthma-related allergens, such as house dust mite. So for example, in HDM, there's multiple proteases, TRP1, TRP3, TRP6, actually TRP9 as well. So again, in other allergens, and this already being linked to the inflammation, airway epithelial inflammation, asthma. So we thought maybe we should look at whether the protease in this allergen extract could potentially cleave gastromin B. So this is an experiment that either in ABC use the lysate from 293 T cells transfected with gastromin B, so full length tagged, and an increased amount of the HDM extract, you can see actually lead to the pretty nice cleavage, not just total degradation. Actually, it's looks like very specific in the cleavage at a certain site, because it gives you a distinct band. It's time course and other extract like the Alternaria extract, cockroach extract, also lead to the pretty defined product of gastromin B. And then in the HBE cells, that two donors, if you incubate with HDM, you can see this very nice band of gastromin B being cleaved. So where does HDM protease cleave gastromin B? So this is a site we already tested for caspase-1. We know this is a caspase-1 site. So again, this shows that this site in caspase-1, the D236 is a site, but here, even in this mutant gastromin B, it still gets cleaved. It doesn't care actually. So this suggests that actually HDM cleave gastromin B at a different site from the caspase-1. So this experiment we did. So we want to determine the cleavage site. So this is we get the recombinant gastromin B, and then if you incubate with HDM protease, it gets smaller band, and then this HDM protease alone, we just run this. So we know this band is because we are looking for the C terminus of gastromin B, and we know that around the size of 15 kilodalton, and this one is a protease alone. So we specifically cut out this band. It's not here, so means that this is most likely coming from gastromin B. And then did the protein sequencing. I think most people now do not know actually that you can actually sequence the protein. So here we look at the N terminus of this C terminal fragment. So you can sequence the protein. There's not many people doing that actually, but when I was grad student, actually, I used one of these methods to determine one of the proteins. So I know the method, but again, this was done by core through the service, and they can determine the sequence of protein. As you can see, some they can, because the HPLC peak, there's multiple peaks, they can know it, but sometimes they are pretty certain that, for example, this one definitely is in methionine. So you can pretty much from this you can match the protein sequence. In fact, if you look at here, it's beautiful. Like, I mean, every time you see this one, I got excited because it's clearly serine, leucine, glycine. So you can look at here actually mapped so nicely to the gastromin B, and we know it, but every time you see it, you still got like some goosebumps here, because it really show that this C-terminal fragment, the N-terminal starts here. So this is the cleavage site after this lysine. So again, this is a gastromin B, this is the cleavage site. and cleave into this slightly larger protein, 244 amino acid. If you express this protein in the cells, massive apoptosis, again, you can see here, this is a previous cleavage by the caspase-1. So that paper was published in last year in Allergy. So now, if you look at the pathway, rhinovirus is an asthma trigger, can induce expression of gastrin B. By the way, I should mention that Xiaobo Zhou and Benjamin Raby's group actually published a really nice paper earlier this year showing that rhinovirus and interferon in particular actually can really jack up the expression of gastrin B. So you can read that the published in ERJ. But our work also showed that the house dust mite can actually specifically cleave the gastrin B here. So two really important asthma triggers act upon this pathway. Okay. So we believe this is a very important, probably not the only pathway, but it's a very important pathway in asthma pathogenesis. So can you actually do something about it? As I mentioned, there's no cure. We're not suggesting there's a cure, but can you do something potentially therapeutically on this pathway? By particularly inhibiting the gastrin B, we know if everything is because of the apoptosis, can you inhibit this apoptotic activity of gastrin B? Again, this is an amazing work by Ron Paginiban. What he did is he transfected the cells with gastrin B N-terminus, okay, very quickly. So this is called reverse transfection. Basically, allow you to do a high throughput, large scale transfection, and then he played the cells here, and then screen against the library of small molecular compound and added very quickly within an hour of transfection, because you don't want this to sit too long because they're going to die. So idea is that you identify some inhibitors of this apoptotic cell death, okay, through this just measuring the LDH in the cell medium, and then you can identify the small molecule hits and validate them. And this work was done at the Harvard ICCB Longwood screening in the facility. I'm not going to go through the LDH assay. Here is a typical screen you get. You have this, each of these is experimental compound. You have negative control DMSO, they don't change much, and positive control here that can block this cell death. And then you can screen, look at the potential compound from library that also, like positive control, can inhibit the cell death as measured by LDH. We screen about many libraries together. There are about over 23,000 compounds we screen. And this is the distribution of different libraries. Some libraries are smaller, for example, this GPCR library, this the biolipid library, the Cambridge library has the most compound here. So this is just the distribution of the compound. So this is data from all library screen. So this is most of compound here. Obviously, we look for any changes here. So the blue is we call the strong effect, the medium effect, and then there's some weak effect. We did a lot of statistics because of calculate the score, you know. So this is the distribution of hits by libraries. Together we have from the pyramid screen 171 positive hits. We test the 80 of them because some compound we just couldn't get it for rescreening, and then we validate most of them, and then we look for the compound that can act at the sub-nanomolar, sub-micromolar range. So here at the nanomolar range. So together 10 compounds. This is a list of compounds we have, and if you look at them, some of them actually the top two are polo-like kinase inhibitors that have been designed to inhibit the polo-like kinase. We don't know how this compound will inhibit the apoptosis activity of gastrin B. But these are and also this famous red wine chemical that we also shown that it might have an impact on the gastrin B apoptosis. Again, we have not characterized any of them carefully, so this is a pretty preliminary data we generated. So I want to summarize here what I told you today. is that in the first story, I showed you that we identified a coding variant, a splicing variant in gastromin B that associated with asthma. That variant can lead to the change, a skipping of an exon, and that is eventually abolishing the ability of gastromin B protein to induce pyroptosis. I showed that several asthma triggers that can act upon this pathway. Rhinovirus infection can increase the expression of gastromin B gene. Allergen proteases from extracts such as HDM can cut the gastromin B at a very specific site, releasing the pyroptosis-inducing N-terminus. And in the last sort of more preliminary study, we identified a few small molecule compounds that may block gastromin B-induced pyroptosis. Some future directions. So really, I'm very interested in the HDM, the other allergen extract. We want to identify what the specific allergen protease, because there are so many of them that can specifically cleave gastromin B, and then they're what kind of proteases, right, can cleave gastromin B at such a specific manner. And one thing that we, and actually many reviewers also want us to show that whether this gastromin B cleavage and hopefully the pyroptosis will actually occur in vivo, obviously in human airways, in transgenic mouse airways. Again, as I mentioned, there's no gastromin B homologue in the mouse genome. I mean, that might be one of the reasons why initially nobody is looking at the gastromin B, because there's no mouse homologue. In fact, they don't have a functional homologue even in non-human primates, in monkeys. The organisms that express gastromin B include horses, which we know that asthma exists, and then cats, maybe dogs, I forgot. But we look at the monkeys, there's no homologue. We look at the horses, there is a gastromin B. So anyway, so we want to see whether this actually phenomenon we observe would actually occur in vivo. So, and then thinking about therapeutics and whether we could expand the small molecule screen and identify more compounds that we can test in vivo to see whether they can block the allergen-induced gastromin B-mediated pyroptosis. Finally, I want to thank the people who did the work. I already mentioned Ron Panganiban, highlighted here again. He's an amazing postdoc, and I should say that he right now, last year, he got his independent job at the Cincinnati Children's Hospital, and his lab is also interested in gastromin B and other gastramins in allergy and asthma. And Mao Yunxuan contribute to the work. Again, here, other people in my lab, and I want to thank my collaborator, Jenna Park at the School of Public Health, who provide really amazing RI culture work that enable our studies. And then Scott, Mike, Elliott, who I collaborate early on in my career, Kellen and Blanca, also on the asthma work. My funding, obviously from the Heart and Lung Institute, supported my asthma work. Also, I did not talk about, but the American Asthma Foundation supported some work in the airway smooth muscle cells. So I would stop here and happy to take any questions. Thank you.
That was can you hear me?
Yes.
That was a wonderful talk, Chen. Really wonderful work, and we have several questions in the chat, and I would encourage other people to put questions in the chat. So the first question says, great presentation. Which region of the N-terminus is missing in the rare variants to not allow the induction of pyroptosis?
Yeah, so N-terminus. Oh, I just terminated my slide presentation. So exactly at the very end of the N-terminus. So it's the C-terminus of the N-terminus fragment that's missing, certain amino acid. Yeah, it's critical actually. In the structure, you can see why that region is important, because if you don't have that, the pore cannot be formed.
Okay.
In fact, the 2020 the science paper last year talked specifically about this this splicing variant. They actually have a structure of the 416 and the 403, and it's very clear from the structural work why that region is critical for proptosis.
Okay. Next question says, thank you for a fabulous talk with superb clarity. I haven't usually thought of epithelial cells as the major source of inflammatory mediators in asthma. Your discussion of allergen protease and gasdermin B seemed to bypass IgE, mast cells, and type 2 inflammatory mediators. Do epithelial cells play a dominant role in asthmatic inflammation?
Yeah, so the IgE, so the gasdermin B, that 17q21 association, if you look at the initial the association actually associated with mostly with childhood onset asthma, not IgE like allergy type of asthma, not IgE dependent asthma. So I don't think this this only this the impact on IgE asthma we don't know, but this mostly again the childhood asthma. So the cytokines that most people study in the proptosis, obvious the IL-1 beta, IL-18 now a little bit. So yeah, in that part I'm not the expert, so I can't speak, but I know that these pathways will probably have lesser importance for IgE type of allergic asthma.
Mm-hmm. Okay. So, and if I could follow up on that question, so when the very interesting data on the proteases and the house dust mite proteases, we normally think of house dust mite induced inflammation and its contribution to the asthmatic diathesis as being one that's related to allergic responses. And so, and this follows up a little bit with the prior question. Are you aware of any crosstalk here between what's happening?
Yeah, I mean,
as
the
well
obvious there.
and,
Yeah,
and
there's a lot of
induction allergic?
the
Yeah.
mast cells, other types. We have not looked into that. I think that one of the questions people always ask is that that, for example, how yeah. So for your specific question, we don't know the interaction. I assume probably the house dust mite is not just to act through their protease. Other components, even the protease, probably will do damage to other maybe recruit other immune cells into the site. So got to be an interaction between the epithelial cells and other immune cells. So on that I don't know specifically, but for the how actually protease from the house dust mite, because it's extracellular, right? So
Right.
to act on the gasdermin B in the epithelial cells, the protease need to actually get into the cytosol of the epithelial cells. So therefore, that really, how do these proteases get endocytosed and become functional in the cytosol is a major question. That's why we have done work try to incubate, for example, the ARI HBE cells with a protease extract. We don't see that unless you break down the cells first. So there's got to be some kind of compromised epithelial cell, integrity for the protease to get in. So maybe that's where the inflammation, other immune cells comes in.
Another question. So you showed very nice work about trying to screen for potential inhibitors or blockers of gasdermin B. There's been a lot of work now of people talking about how we don't need to do screening and we can just use AI to
Yeah.
look at these structures and come up with competitors. Have you thought about doing that? And have you compared that to what you've seen in your screens in terms of thinking about ways to make progress in that area?
Yeah, I'm pretty sure a lot of groups are looking at that. We're not expert in the structural biology or even all this design of, I think that gasdermin D, definitely, I know groups are doing that because that's a major, more canonical gasdermins. They had a structure earlier, so they must have that work going on, but I haven't seen anything published yet. Gasdermin B structure was solved last year. You saw the nice pore formation. So from that, you probably can, computer AI, computer is powerful enough, potentially can predict what kind of compound they can design to disrupt the formation of the pore. So yeah, but not from my lab. Yeah.
Okay, all right.
So these are really exciting possibilities.
Yeah, yes, yeah, I think so. I was there aren't any other questions in the chat. Again, I wanted to thank you for a really wonderful talk. I think people are seeing, I hope seeing the schedule of our next set of asthma talks. On my screen, it's actually being overwritten, but is that showing up?
Yes.
Okay, so you can see the next sets of talks, so we're really looking forward to talks that we hope are just as good as what Chen has given us. And the next talk is going to be mast cells and asthma, more complicated than we thought, from Josh Boyce, and that's in November. And then in January we have airway smooth muscle pathobiology and asthma exacerbations, and that's by Ramaswamy. And unfortunately, I can't read this. Ramaswamy Krishnan. And then lastly, and then in March, the role of viruses in asthma development and severity by Peggy Lai. And then lastly, an update on pulmonary function test interpretation by Nancy Lang and Barry Shea. And so an exciting set of talks. Thank you again, Chen, and thank you everybody else for attending. Remember, please put it write to Israel ASST at partners.org if you'd like to get CME credits. And we hope to see you multiple times in this coming academic year. Have a good day and have a good weekend.
Thank you. Bye-bye.
Bye-bye.
Bye, Chen. Great, Chen.
Yeah, that was wonderful. Thank you so much. Okay, Chris, thanks so much.
Thank you. Thanks, Franco.
Yeah,
Thank you.
Franco.
Good talk. Good talk.
Yeah,
Good.
great.